Okay, welcome to another edition of the Mariner Biology YouTube channel. This time we're doing something for AP Biology, and we're going to be talking about how to interpret the results of a gel electrophoresis we did in our big biotechnology lab. So, we were given two plasmids, and the names of those plasmids are pKAN-R and pARA. So remember that plasmids are small circular pieces of DNA that are found in bacteria in addition to the one large circular chromosome. Okay, and the pKAN-R plasmid has a gene for kanamycin resistance and a gene for the red fluorescent protein, which is the one we're trying to get into the bacteria and eventually make them fluoresce red, give them a new trait. And the pARA plasmid is a plasmid that contains a gene for ampicillin resistance. It contains a gene for a regulatory protein called ARA-C, and then it also contains a portion of the arabinose operon that we call the promoter. Okay, and we're going to use that promoter for a very specific purpose. So here's our two plasmids, and you can see the genes that have been engineered into these plasmids. Okay, and what we're going to do is we're going to cut these two plasmids with restriction enzymes. There's two different ones. We're using Hindi3 and BAMH1. Okay, and each plasmid has a site where these restriction enzymes are going to cut. So let's check out where those are on the other one here. Okay, so remember that we had four tubes, and there was a K plus and a K minus tube. The K plus has the pKAN plasmid with restriction enzyme added to it, the two that you see here, and then the K minus tube has the plasmid without any restriction enzyme. Then we had two other tubes, the A plus tube that had the pARA plasmid with the restriction enzymes added, and then the pARA plasmid with no restriction enzymes was in the A minus tube. So we have K plus, K minus, A plus, A minus. And what we end up with when we digest these two plasmids in their separate tubes with the restriction enzymes is this. Okay, what happens is the two restriction enzymes cut on either end of the RFP gene, and so we get one long piece that contains the kanamycin resistance gene, and then a smaller piece that contains the red fluorescent protein. In the next plasmid, the pARA plasmid, we also get two pieces, but one of the pieces contains all three genes, or actually two genes in the promoter. You got the ampicillin resistance gene, the ARA-C gene, and the promoter, the pBAD promoter. And then we get this smaller 40 base pair chunk that contains really nothing of interest. Okay, and so what we end up with when we digest with these restriction enzymes are sticky ends. And what we're hoping for is to take the larger piece from the pARA plasmid and have it combine with the RFP portion from the pKAN-R plasmid. So we get a new plasmid with these two parts. And the other ends are also going to have sticky ends, so we're actually going to get all kinds of combinations of stuff in the tube that we add all this to with the ligase to try to get all this stuff joined together. So what we're hoping for is that we get the two pieces that I've drawn here with the sticky ends to actually join together and form a new recombinant plasmid that we can then get into the bacteria and have the bacteria transcribe and translate the RFP gene, and actually we can confer a new trait on that bacteria and hopefully see it glow red. So what we need to do is confirm that we actually got this product, and we do that with gel electrophoresis. Okay, so here's our gel, and we're going to draw in our wells. Okay, and we've got what we're loading into each of the wells. So we're loading a base pair ladder, and we're going to use that for comparison because we know what size the pieces we're looking for in each lane are. So if we see fragments of those sizes, then we should be good to go. Okay, so remember we've got a negative end, a negative electrode, and a positive electrode, and DNA has a negative charge, so as we flood that one end with negative charge, it's going to push the DNA out of the wells and towards the positive end of the gel. Okay, so we'll see what this ends up as when we run the base pair ladder. Okay, so there's all our fragments of different sizes, and we're aware of those sizes. Okay, and then if we look at the first lane, the K plus lane, there's our two pieces, right? We've got the one piece that represents the larger chunk with the kanamycin resistance gene in it, and then we've got the smaller chunk that just has the red fluorescent protein gene. If we look in the next lane, the K minus, right, that plasmid should have stayed intact, and you'd expect that there'd just be one big band for that one plasmid, but we actually see multiple bands sometimes, and that's because the plasmid can behave in different ways. We might have two plasmids actually joining together. We may have a plasmid actually supercoil itself, and we read about that in the introduction, that when the plasmid supercoils itself, it actually kind of tightens up a little bit and it can get through the gel a little faster. So we've got those things labeled. Okay, so that's what we're seeing there. And then in the next lane, right, the A plus lane, now we've got this fragment with the ampicillin resistance gene, the ARA-C gene, and the PBAD promoter. And then way down the gel, we've got that tiny piece, that 40 base pair fragment that we don't really care about, but it shows up down there, and we can see how a smaller fragment can get through the gel a lot faster. Okay? And then, so what do you think you're going to see in the A minus lane? What would you expect? Would you expect that one band again because it's only got the plasmid in it, or are we going to get the same thing that happened in the K minus lane? And it turns out we're most likely going to get more than one band and get these different products because of the behavior of the plasmid itself. Okay, and then when we add the tube that contains everything that we added the ligase to, we end up with something like this. Okay, so there's our ligated plasmid, and remember that we've got all those chunks that we've digested floating around, so we can get all kinds of combinations of things, and it's possible that something else that then, besides what we were hoping for, or in addition to what we're hoping for, could end up in that lane, okay, and in that tube. So that's pretty much it. I hope this kind of gives you a little bit more background on what we were looking at when we actually were able to stain those gels and see our bands. And if you have any questions, you can ask me tomorrow in class again, but please give this a good look before you do. Thanks a lot. Have a good night. Bye.